welcome Dr. Aziz El AMRAOUI, who is working in Paris. He is research director at the PASTEUR Institute. And dear Aziz, you will present the work of your team regarding the pathophysiology and therapeutic option regarding progressive sensory disorder. Thank you, dear Aziz, for being here today.
Thank you. Hi, everyone. Good afternoon. So here is the title and some outlines of my presentation, but I would like first to thank Carol and the organizing committee for the invitation, and also for the programmation, because we had very, very exciting talks, and the nice talk this morning by Jonathan, Didier, Fabrice, and just now by Jean-Luc. And even some of the questions this morning about evolution, the question about tinnitus, and the answer nicely, in fact, set the stage for my presentation. My presentation will be about pathogenesis and therapy and how these are connected. In fact, like for any organ, the inner ear included, the disease or the deficit is just the dark side or the mirror image of what should have been there, what should have been normal. And the balance or the change between to one path or the other is governed by genes and what we experience during our lifetime. So gene and environment are two small words, but in fact they trigger an infinite world. We may still have, as Jonathan mentioned this morning, we are not far from having understand all what's behind hearing loss. So we may still have next 100 years to understand how gene and environment shape our destiny. So this is important because in hearing, we know they are frequent, they are highly prevalent, but we use the word deafness, which is somehow singular, la surdité in French, but in fact, they reflect hundreds, if not thousands, of different situations. This is correlated with the question about tinnitus, and in fact, what we have are more likely deafnesses, and we should always talk about deafnesses and the surdité or multiple tinnitus. This is important in terms of therapy or treatment because in an ideal world, an ideal treatment is a treatment that will deal with the real and primary cause of the deficit, not its consequences. It's like now in this pandemic, you are given the choice to choose between the vaccine that treat the cause or hydroxychloroquine that may or may not treat or fight against the virus. This is important because, as I mentioned, there are not one deafness, but hundreds. In fact, there are thousands of causes, different structures, different pathways, mechanisms behind hearing loss. And here in clinics, you have two audiograms, one to your left that reflects profound deafness for all sound frequencies that can happen at any time, during lifetime. And to your right, you have one for age-related hearing loss. But each of these audiograms can be observed in hundreds of patients, each of the patients having a different cause behind that audiogram. And unfortunately, in humans, we do not have a lot of additional data. We have some nice presentations by Fabrice and Jean-Luc trying to find and develop new tools that may help add some additional information that can guide us towards trying to find more information about what might be the mechanisms behind the disease. So trying to understand the causes behind the hearing loss is trying to find which of the structures from the periphery, the external middle ear or the inner ear within the hearing organ that we see to your upper top right, Which cell type? Is it supporting cell, hair cell? And when you identify the cell, in which organelle or substructure? Is it the apical hair bundle? Is it the synapse? Is it the fiber spontaneous rate, low, high? Is it beyond the inner ear? Is it central? So these are questions we need really to answer if we want to treat real cause behind any situation for hearing loss. That's the goal, but we still have many, many years to reach it. I have just one slide about the anatomy, thanks to previous talks by former speakers. Just to highlight some structures I will be talking about much later. As we know, sound comes through the different part of the ear, reaches the hearing organ. This is a picture from Promenade autour de la cochlée by Jean-Luc Puyel's team. I was happy that Jean-Luc described the difference between outer hair cells, inner hair cells, because that will be important also during this talk. These cells are where, in fact, sound is transduced, transferred into electrical signal in this highly organized structure, which is called the hair bundle, containing stereocilia organized in staircase pattern. Once the cells, and in particular in the inner hair cells, are stimulated, you have glutamate release, and talk of Didier nicely detailed its calcium control of the release, glutamate release, and the importance of having the calcium channel at the active zone attached to the ribbons. All these informations are important for in healthy situation. We will see sometime later what happens in some pathogenic conditions. Another point that I want to highlight here, which explains many of the challenges we are facing in the audition world, is evolution. Usually underestimated, but it's really important, and we will see how. Mechanoreception, I mean the deflection of the hair bundle and conversion of sound, is called mechanotransduction. And mechanotransduction is present almost in all kinds of species. But hearing is a late invention during evolution. Real hearing starts after fish. And this slide was a better illustration of how the inner ear evolves during the last 500 million years. In fact, the inner ear originally is a balanced organ. It's composed only of vestibular sensory epithelia, and it's some hair cells within one of the vestibular organs that start to perceive acoustic stimulations. But only after we move on from water to in tetrapods, with the appearance of amphibians, we start to have a real organ here indicated in red, real organ dedicated to hearing. This organ starts to elongate, to further elongate, forming this basilar papilla in reptiles, chicken, and the spiral cochlea in mammals. There has been, over this 500 million years, many innovations to enable us to hear and communicate. And among the changes is the transformation of the hearing epithelium from this standard supporting versus hair cell organization to a highly complex organ with different supporting cells, and the appearance of these outer hair cells that we found only in mammals. My take-home message here is that we, as mammals, in our inner ear, we have the vestibular hair cells, the inner hair cells, and the outer hair cells. And these are completely different cell types. There are, if we can say, ontogeny reflects phylogeny. In our inner ear, we have cells that reflect 100 million years difference in terms of evolution. And we will see how that impacts treatment. One additional impact is that during this million years, from fish to mammals, we lost progressively, as Jonathan Gale explained this morning, the capacity, or the hair cells have lost the capacity to regenerate. Regeneration, which is spontaneous in chicken and induced in other species. That is also important, one of the explanation of why deafness and hearing loss is highly frequent in humans, because any damage is irreversible. This impacts not only the prosthetic treatments that are now available and their performances, but also progress in the clinical therapies that are underway. And now I will be moving to explain situations where how we can move on from pathogenesis to treatment, and how we can try using new therapies, especially gene therapy, try to restore the specific pathogenic abnormalities that are seen in pathogenic conditions. So for my next part of the talk, I will focus on mouse models, because thankfully the inner ear in mouse work much the same time as in humans. So we use animal models to try to determine the precise sequence of events that occur in the defective situation for a given gene of interest. For the next slides, beyond, in fact, the name of the gene, what will be important is the situation that will be illustrated in the mouse models, where I will try to give indications, or at least informations on different types of situations. One where we have a congenital form of hearing loss, another where we have late onset and progressive hearing loss, sometimes combined with the appearance of other phenotype, notably the balance deficits. So my talk, or my next slides, will be about syndromic deafness, which is called Usher syndrome, the form type 3, which is characterized in humans by postlingual hearing loss, which is progressive, and variable onset and intervals of severity of balance and vision abnormalities. Just here in the bottom, you have Usher 3 defected patients. They have all the same age, but one who have profound hearing loss here to your right, in the middle moderate hearing loss, and mild hearing loss in another. So even sometimes for the same gene, we may have different phenotype expressivity. So to study this human deafness form, what we did is to try to model it in mice. And in the lab, in the team, we have the capacity to create mice, total knockout mice, where the gene may be inactivated in all cell types and from the early stages of embryonic development. But we have also the tools to specifically inactivate the gene only on some cells, notably the hair cells, and only from birth onwards. So for this gene that is expressed both in neurons and hair cells and from embryonic and adult life, it was important to discriminate what's the part due to embryonic development, what's the adult function, key function, and which cell type is important. Now, if we look at the hearing sensitivity in these two kinds of mice, in digital mice, that is always will be presented in blue, as you can see, the EBR threshold are over 100 decibel, which indicates complete profound hearing loss in these mice. And this can be observed as soon as the mice start to hear from 2 to 3 weeks of age. Now, if we look at the in the conditional knockout mice, where gene is inactivated only in hair cells and only after birth, you can see here in red the hearing threshold that is about 20 decibel after 3 weeks, and the severity which progresses over time with 100 decibel by 2 months of age. If we try always to try to extrapolate the data from mice to humans, and in this situation, what we saw is that it's the conditional knockout mice that better mimics the situation in humans, where hearing loss appears after acquisition of language. On average, the hearing loss or the start of hearing abnormalities start to be noticed by the age of 10 years of old. Now we move on to look what's behind these different hearing sensitivities. What happens at the level of the morphology of the organ? These are things we can never do in humans, at least the imaging technology that we have today cannot allow us to reach the degree of high resolution needed to check what happens at the cellular level. Here in mice, we can check what happens in the hearing organ, and you have a control mice to your left. In the middle, in the total knockout mice, you can see that the hair bundle is completely disorganized, and even first row of the stereocilia is completely missing. And this totally explains the profound hearing loss that we can see in these mice. Now, in the red mice, the conditional mice, where the gene is inactivated only at postnatal stages, the hair bundle is completely normal, at least in terms of morphology, and all the stereocilia are there. But still, this mice has hearing deficit. So we have done additional tests in collaboration with both Paul Avant from Clermont-Ferrand, Fabrice Giraudet, and also the team of Didier Dulon in Bordeaux. And what we found is that these mice, in addition to potential hair bundle abnormalities, they have also deficit in neurotransmission. Here in red, you have the current, the calcium current in the mice in absence of this protein, which is called clarin-1. What we find is that in absence of this protein, you have more calcium currents, but the problem is when you go to look at the morphology here to the bottom right, you can see that the calcium channels here in green, there are more than normal situation, but unfortunately, most of the channels are outside of the active zone. So what's important is not only to have the calcium channels, but you need to have those calcium channels organized in a nanodomain organization just under the ribbon where they are supposed to act. So this is, now we have a clear picture of what happens in the pathogenic situation. Now, in terms of treatment, the question, what can we do in terms of gene therapy? Can we restore these abnormalities? Can we restore all what we see in these mutant mice? So here we use, in this work in collaboration with Said Safiuddin, gene therapy. We use adeno-associated viruses having an intact clarin-1 that we inject through the round window in the cochlea. Here are just the name of some of the people involved in the work. So what we saw is that after one injection, if we did one injection at just the first day after birth, if we do that in the total knockout mice, 3 weeks after, we see 10, maximum 15 decibel of restoration, not too much. In these mice, if we continue and try to look what happens at the level of the morphology, as we can see here, with or without the injection, the hair bundle is still completely disorganized, meaning that this injection didn't allow us to restore the normal shape of the hair bundle of the sensory hair cells. Now, if we do the same thing, the same injection, the same date, but in the conditional knockout mice, the red mice, you can see that 3 weeks after injection, here in this purple curve to upper right, we completely restore the hearing thresholds. The injected mice behave exactly like the controls. And this restoration is stable also over time. Even at the level of the morphology, unlike here shown at 4 months, unlike in the pathogenic conditions where we completely lose the hair bundles and even the hair cells and the outer hair cells and at the level of the inner hair cells, the one injection allowed the organ to persist and resist against degeneration. That was about what happens at the apical part of the cell. Now, if we go, if you remember, I also mentioned neurotransmission deficit in both mutant mice, total and the conditional. one injection in both mutant mice, in fact, allowed us to restore all the synaptic features that was found in absence of the protein. Both the exocytosis, glutamate exocytosis, its calcium control, and even the localization of the calcium channel, as you can see here, the green staining, which is no longer expanded beyond the active zone, but completely attached to the synaptic ribbon. I still have 5 minutes. I now mentioned what happens in the hearing organ at the cochlea. I have 3 slides about the vestibular part. As I mentioned, the H3 patient had also balance deficit, and this can be also reflected in the mutant mice. Balance deficit in mice can be followed both by following the exploratory behavior in the cage, or in the platform, how much time is spent in the platform, or using the swimming test. And we have also other different kind of tests. And as we can see here, there are some situations where the mutant mice has a completely bilateral vestibular deficit with this typical circling behavior. Now, the same injection we did in the hearing cochlea, in fact, in these mice, can completely restore the vestibular deficit that we see here in the upper slide. This is a mouse that was injected at P1 and observed 2 months later. It behaves like a normal mice, both in the open field, but also by measuring the VOR, which is the vestibular ocular reflex, swimming test, rotarod, etc. So to conclude, based on this model pathogenic conditions, which shows congenital versus late onset with or without vestibular deficit, what we have seen is that one injection can allow us to prevent abnormalities that occurs afterwards, as what we have seen in the conditional knockout mice. But in some situations, one injection afterwards cannot restore what occurred already before birth, especially if it's about the structure of an important organelle like the hair bundle. The other conclusion was that our gene therapy show us that synaptic transmission or the signalization deficit that we see in the 2 mutant mice can both be restored with the one injection, one postnatal injection. And as we have seen, the vestibular deficit can be totally prevented by gene therapy. So gene therapy, depending on when it is applied, to which it is applied, is working. We have now clear indications that in mouse it's working. So one of the next question in the future, and say it will follow up on that, is how can we move on from mice to humans? I think we still have time for that, but again, there is no miracle treatment that can be applied to all forms of hearing losses. A treatment, even a gene therapy, will be on a case-by-case basis. It will be adapted to each pathogenic conditions, and what will be the target? Is it the vestibular hair cell, the inner hair cell, the outer hair cell? Depending on what's the question, the response will be completely different. And the question will be also what type of signalization or deficit. Is it structural or is it a signalization pathway? And the intervention, when the intervention occurs, is it before or after the symptoms are there? All this will be important, or how we will answer these questions will be important to accelerate what we have now in preclinical animal models to move on to humans. And this, of course, will require contribution not only by scientists, but clinicians and all actors in the audition field will be needed to speed up the process. Now, just this is my last slide to acknowledge the people in the team who contributed to the work, the funding, and also the collaboration I mentioned. Steve Brown and Mike Ball for some of the mice, Said for gene therapy aspects, Didier Dulon and Paul Avant for some physiological measurements along with Mathieu and François. Thanks for your attention.
Thank you very much, Aziz. I'm sure there are some questions for you in the audience. Who would like to start with the first question? Patrick Randon.
Thank you for your very nice talk that gave a new clue for molecular aspect of deafness. But I have two questions. First, do you know, you say in the first part of your speech that during evolution, the cell of the inner ear cannot regenerate. Do you know if there is stem cells in the cochlea, and if the cells are different, and if you have a marker? This is my first question. The second question, in deafness, there is maybe the role of prestin, which is another protein along those you speak about. And so I want to know if you can combine your approach with these other proteins that can be involved in deafness. Thank you.
Okay, thanks for the question. And to answer the first one, which is about evolution and linked to some of the talks this morning about regeneration. As I showed in my slide when I mentioned the evolution of the inner ear from fish to mammals, I have shown also this row that shows that over this millions of years, we start to lose regeneration. And this is due to the fact that over time, there are less and less stem cells that will allow us to trigger new divisions and production of new hair cells. So there are stem cells or supporting cells can re-enter this division cell cycle and create new hair cells in fish and also in chicken. There are some indications that we may still have some few stem cells or ability to regenerate hair cells, especially in the vestibular system in mammals. Some have tried to see, or at least in vitro, can show that even in the cochlea, we can have indication that there are some. But again, and it was mentioned this morning, you may still have some capacity to create new hair cells in the mammalian hearing organ, but only at the earliest stage of development. Once the cochlea, for me, once the cochlea is there, once you have all the hair cells set, we have lost all the signalization that will allow us to recreate new hair cells. But most of the work that is done now in chicken is try to find what are the key pathways and how can we reactivate those lost pathways in mammals. That's part of, for instance, Jonathan Gale work and others. Yeah, regarding the other question is about prestin. Prestin is a protein that is the protein that is involved in outer hair cell amplification. It's just one protein. It's important for the outer hair cells, but in most of the other deafness cases, prestin is not mutated. I mean, even if we bring it in, it might not add something else. Gene therapy for prestin will work for people who had mutation in prestin. That's why when I said gene therapy, once we have it, it will be on a case-by-case basis. Gene therapy for prestin will be for prestin. Other gene therapy for another gene, we have to adapt it and prove that it's working.
Thank you.
Thank you very much. Is there any other question for Dr. El AMRAOUI? I do not see anyone raising his hand. In this case, I will thank you once again. Yes, Jonathan, you have a question.
Yeah, that was great, Aziz. Thanks very much. I just wanted to give you an opportunity to speculate on the possibility of doing in utero kind of gene therapy in people. I mean, you know, I know that's really difficult, and you've alluded to the fact that this is where we've got to go, but I just wondered your thoughts on it right now in terms of that.
Yeah, that's a good question. And I think Saeed will be addressing that question right after in his talk. But again, you raise one important question, which is the fact that we are using mouse models. And we all know that mouse from P1 from birth to 3 weeks, in fact, corresponds to 5 months to 7 months of gestation in humans. So, which means that one of the key questions that will be once we have a gene therapy is to prove at which time it's doable and beneficial in humans. That's why also I mentioned in my conclusion the importance of preventing instead of correcting, meaning that by focusing also on late onset forms of hearing loss, we still have the therapeutic window where we can intervene without having to go in utero and try to do something which is ethically and deontologically much complex. And I would say for now, forget it. Thanks for the question.
Thank you. And Carol Baron, you have also a question for Aziz.
Yes, I have a question, a general question. I wanted to know, you were saying that Usher syndrome leads to deafness and blindness, and is there a link also to dementia?
For Usher syndrome, no. Although we know that some of the genes may have some function also in the brain, but for me, I think even that function they still have in brain may be compensated for when the gene is affected. Meaning when you have a mutation on the Usher genes, the key organs that are where you have a pathogeny will be the peripheral inner ear, and that results in either hearing or balance, and also in photoreceptor cells of the retina, which is why you have a vision deficit. But all Usher patients, they can have normal life, no dementia, no problem in terms of cognition or cognitive deficits.
Thank you.
You are welcome.
Thank you. I think we have no additional question for Aziz. So I would like to thank you once again, Aziz, for your presentation.